Welcome back to part two of the third evening of Wonders of the Salish Sea. We are on to our second presenter, Josie Islin, and she is a photographer, author, and designer of many books, including The Curious World of Seaweeds. Josie holds a BA in Visual and Environmental Studies from Harvard and an MFA from San Francisco State University. Her prints speak widely on the confluence of art and science and advocates for ocean health through education, activism to preserve seaweeds and kelp forests of our Pacific coast. Over to you, Josie.
Great. Well, thank you, Liz, and thank you everyone there in the Salish Sea. I'm actually chiming in here from San Francisco, from the ancestral lands of the Ohlone Indians. But the seaweed flora that I'm going to talk about really extend up through Northern California, Oregon, Washington, and up the coast to British Columbia and into the Salish Sea and Puget Sound. So it's these cold North Pacific waters that are so important. And I'm going to share my slides. And what I'm going to do is take you on my journey as an artist into the science of seaweed. I have been using my scanner as a camera since about 1995. And it has allowed me to create these portraits of the marine algae. And so this will be partly my exploration as an artist, but also I will be bringing, interspersing information about the seaweeds themselves, which of course, unlike the whales that we just learned so much about, which was so cool, these are the primary producers. This is the marine algae that is at the bottom, that creates the bottom of the food chain and creates so much habitat for so many organisms. And so in about 2008, 2009, I started thinking about the marine algae and realizing that, while there are cetacean societies in all the 50 states probably of the United States, there are no kind of popular algal societies. And if you have presentations on whales or sharks, it is sure to be extremely popular. But the marine algae, the seaweeds, they need their stories to be told. So I am really the crusader for the marine algae. There's a bunch of us. There's an incredible group of scientists called the Phycological Society of America, the PSA, which is phycologists, or what seaweed scientists are called. And I've learned so much from them. So this is a project that is in a gallery in Alameda, and what I found is that I can really produce these images of the seaweeds that I can install in all these different situations, whether it's a fine art gallery or an architectural or a public space. This is in a gallery space, and so these are 94-inch tall curtains, and what you have is the rosy red Erythrophyllum on the left. You have the Agresia menziesii, or Featherboa Kelp. Next, going from left to right, then you have Palmaria, which is the West Coast dulse, is a common name for it. And then on the right, you have the Macrocystis, or giant kelp. And as I go along, I'll kind of talk about the basic kelp forests that we have here on the eastern side of the Pacific Ocean. So this is Macrocystis, or giant kelp, and this is an installation in a underused video store and an underused shopping center, where the Arts Commission asked a bunch of us artists to enliven the store windows, and there was a whole festival in this space with lots of kids performances and dances and food trucks, and for me, bringing these algae out into a realm where people get to encounter these organisms that they might not otherwise encounter is really my goal. So this is Macrocystis, or giant kelp, that makes up the dominant kelp forest from Central California to the south. So the famous kelp beds of La Jolla and San Diego are all Macrocystis. And then from about the San Francisco Bay up to the north, up through Washington, BC, and Alaska, the dominant kelp is bull kelp. But the two kelp species intermingle a lot. and their interactions is something that I'm continuing to explore. So how did I start doing this? Well, I happened to be out on one of the reefs. I was learning to be a docent for a reef right near me, to the north of me here in San Francisco, called the Duxbury Reef. And I happened to hold up a scrap of seaweed up to the sky. This was long ago. And I was so flabbergasted by this intensity of color and this fabulousness of form that I knew I had to get these scraps back onto my scanner right away. And so this is my scanner. It's right here in my studio, right next to me. It's a big Epson scanner. And when I'm scanning the seaweeds or capturing the luminosity of the seaweed, I actually use the transparency adapter, which is the top portion of the scanner here with that white cover. So I take that white cover off, and there's a second light element up there in the top. And when I lower the top, that second light element pushes light through the specimen as the scan is being captured. And that is the way that I get these luminous scans or portraits of the seaweeds. You can see on here, I have a Prionitis here on the glass there. Now, what the scanner allows me to do is to scan both small and large specimens at a very high resolution. So I can have all this possibility for how to design with these images, whether it's for the pages of a book, which is usually where some of these projects start out, but there's always the sense that I want to go big. So here again, you have this installation at a shopping center where all this activity was happening. And what you have is here on the left is the Nereocystis lubkiana, or bull kelp. So bull kelp is the dominant kelp in the Salish Sea and around on the west coast of Vancouver Island. Its biogeography, as I mentioned, starts in Central California, but really is the dominant canopy forming kelp that makes up the kelp forest in the cold North Pacific waters along the North American continent, up through British Columbia and into Alaska, and ends in the Aleutians. I really love, as an artist, getting to collaborate with the scientists whose passion for these organisms is so apparent, and I get to really help them kind of broaden their impact, as was the mission for this 2018 conference of the Northeast Algal Society. So a whole bunch of algal nerds, but this gallery space was at the University of New Haven, where the conference was. And I got to enliven the space with these big curtains. And what you have here on the right is one of these big 94-inch tall curtains of a brown algal species called Cytosiphon. And it really has this kind of sausage-like morphology or shape to it. And I was just a couple weeks ago, less than a month ago, I was up in Victoria doing I was with Patrick Martone's marine botany class, and they were out at Clover Point doing a survey, and there was a lot of this Cytosiphon out there. So it can be much larger and more extended in its sausage-like connections, but it's common in the Salish Sea. This is the wonderful Erythrophyllum that I mentioned earlier, and it really is an example of what my scanner allows me to do. It allows me to capture each organism so intently. Not only can I capture these seaweeds and other objects of our natural world with a great amount of resolution so that I can design with them as big as I want or as small as I want, but the color is very spot on. You all know that trying to photograph with a camera in terms of color is all dependent on the vagaries of the daylight or the ambient light, whereas the scanner gives me the correct color. And I have the specimen right in front of me, so I know exactly the color that it's supposed to be. So this again is the Erythrophyllum, the rosy red Erythrophyllum. I'm hoping you guys will all get out into the good spring low tides that are coming around in the middle of the day. And get out into the intertidal zone. This is one you really should find. It really is this rosy red. My friend Patrick that I mentioned actually has this one tattooed on his forearm. He teaches at UBC and has just got an amazing crew of undergraduates and graduate students studying the marine algae of the Salish Sea there. I've done some other more kind of building these more abstracted forms, but always keeping the organism itself super visible. So this is the wonderful Halosacyon or sea sacs. And these sea sacs should, if you are out in the intertidal zone, should be coming up as these fingers sprouting just about now in the springtime along a rocky reef. And they have taken a different tack towards, or different tactic towards survival in the intertidal zone than the more foliose or leafy seaweeds that tend to dry out when the tide is low. So in this case, these sea sacs hydrate from within. They keep whole little universes of ocean within them. And if you find them and you squeeze them, you'll see the water will spurt out the top. And there's some pores up there at the top. I think the Haida word for the sea sacs is the Haida word for nipple. And if you ever find one and squeeze it, you will see why. They are actually in the red category of seaweed, even though they are that chartreuse green. And this is a combination of this very feathery Gloeosiphonia, which is, these were made from two historical pressings from the 1880s. And then I've overlaid color from other algae. But it's to show you this incredible diversity of form and color that the seaweeds present to us. And again, as a designer and an artist, I get to play with these form and color, and it's such a delight. This is a page that kind of brings me back to my roots of Marimekko and design research of the 60s and 70s. So on the left are a series of Egregia pods from our local reef here. And Egregia is the Featherboa Kelp. And Egregia is doing very well in our warming oceans. It is everywhere on the North Pacific. So please go out and find some Featherboa Kelp. And you'll see that the pneumatocysts that kind of buoy the blades up towards the surface when the tide is up, have all of these whimsical blades on them. And I kind of feel like they deserve a tea towel, just like peaches or mushrooms or ferns or whatever you find on your tea towel. So this is the great Egregia menziesii or Featherboa Kelp. And when I was first writing the proposal for my book, The Curious World of Seaweed, I tasked myself with writing an essay called Empathy for a Kelp. And it was the Egregia that I wanted to write about because it was just so foreign from anything that we recognize in terms of plant form, in terms of flora, like say from our backyard. It doesn't have to exert any energy towards woody growth or combating gravity, which we kind of internalize as very necessary. We tend to think of rigid things as more normal. It has these whimsical blades, and it can change its shape, not change its shape, but it can evolve into these very different shapes within the single species. So Egregia menziesii is named after Archibald Menzies, who was the naturalist on board the Vancouver expedition. And Menzies and Vancouver were really in this kind of psychic battle. Menzies was tasked by Joseph Banks to collect as many plants as he could, and they had put this glass house on Vancouver's foredeck to house all these plants. And Menzies and Vancouver hated it. But Menzies had this real affinity for the seaweeds, and he collected these specimens that got sent back to England, that went back to England with him in Vancouver. And then the seaweeds got kind of broken off and given to the expert on seaweeds named Dawson Turner at the time, who wrote a big book called Fuci. So Menzies is a real hero in the seaweed world, and it was your neighborhood where he did all of this collecting, especially, and the Featherboa Kelp is from there. The type specimen is from up there in the Salish Sea, the Puget Sound. And I have really gained lots of inspiration from other artists and started making these more abstracted forms using the blades, in this case of the egregia, as my kind of collage element. And this is inspired by an artist named Rex Ray. And so it's named Rex Ray. I've also been inspired by other artists like Anna Atkins. Anna Atkins was a Victorian polymath who was one of the first people to use cyanotype printing, which was at the time this is about 1840, this nascent photographic process whereby paper is coated with a light sensitive emulsion. And then you can expose this emulsion out to the sunshine with your specimen on top of it. And where the shadow of the specimen hits, the emulsion washes away to this white kind of reverse shadow. So here's that cytosiphon again on the right, made as a cyanotype. And what I realized is that my scanning technique of using my scanner is actually in this lineage of nature printing, whereby the image is made directly from the specimen itself. And it's not mediated by the lens of a camera or the hand of a botanical illustrator. You get what the specimen is giving you. So I started overlaying my contemporary scan onto the kind of more historically referential cyanotype, putting the scan of the seaweed, in this case, Picea californica, into its shadow. And this kind of play, this dialogue between past and present, is for me very much part of my work, and looking at historical context to contemporary issues surrounding our coastal ecologies. And it always has this kind of vector that goes from past to present and on into the future, because underlying all of my work is, where will these organisms be in our changing oceans? So the first book I made was called An Ocean Garden, The Secret Life of Seaweed, and it was published in 2014, and beautiful and very useful in the classroom. It's a visual primer on seaweed, but it promptly went out of print. So I have only about one box left of this book. And then I have to figure out if I can get someone to reprint it. But unlike books on whales or sharks, or as I mentioned, seaweed is a hard sell, and it's gaining. There's so much work being done right now, but I will see if I can find a publisher to reprint it. It's a very interesting situation. But as soon as I was finished, I couldn't. That doesn't stop me from wanting to make another book on seaweed. And in this case, I realized that the history of the science had this visual component to it that was so beautiful. And these are 4 lithographs that I've layered onto this, what is actually the end papers of The Curious World of Seaweed. But these lithographs are all from a publication from 1853 by Franz Josef Ruprecht, who is one of the chief botanists in Russia, German botanist working in Russia, describing the seaweeds of our west coast here. And some were collected in Alaska by Postels, who I'll talk to you about, but these were collected actually down here in California. And you have the Egregia menzicii, or Featherboa Kelp, on the right. And then next to it is the walking kelp, or Pterygophora californica. And then you have Dictyoneurum, which is got this really cool veiny characteristic to it. And then on the left is a Stephanocystis, or bladder chain rack. And it used to be called Cystocyra, if you know the old names. So again, I really wanted to play with my scans working in dialogue with these historical documents. Number one, because it's beautiful, but also because it gets to reference back to how we've learned about these organisms, these incredible kind of primary producers, eco-engineers of our nearshore oceans. And this is Costaria costata, which is called 5-ribbed kelp. And you will find this up in the Salish Sea, on the West Coast, in Alaska. I just saw a picture of a friend of mine holding a Costaria blade in Alaska. which was just enormous. It was as big as her. And what is crucial to remember about these organisms is that cold ocean holds more nutrients than warm ocean. So it's the cold ocean that holds enough nutrients that combined with the magic of photosynthesis lets this kind of robust algal growth take place. So it's really important that we keep our oceans cold for the health of everybody. So this is Nori, or Pyropia, and Nori grows in all the world's oceans. It's very, very thin. It's actually in the red group of seaweeds. So if you're out in the intertidal and you find this very thin, kind of dusky colored seaweed like this, or it could be in strips. I think I have a couple other pictures of it. If you pull it and it stretches, then you know you're in the red category, and it's probably Nori. And this is layered onto a lithograph by Alexander Postels, who was on an 1829 expedition to Alaska and really had an affinity for the drawing and sketching and for the seaweeds, and sent back lots of specimens that then was described by that guy Ruprecht. And Ruprecht and Postels got together and made this amazing folio called Illustrationes Algarum. So here's some more Nori. So what it does is show you this incredible range of the Pyropia or Nori. It can be deep purple, or it can be this kind of dusky green, and you can see the reproductive elements to it on the left here, on the very edges of these folios, these thin blades. So it's only about 2 cells thick, and every cell can photosynthesize, and every cell has access to the nutrients of the ocean. So that's kind of the key to algal growth. Now this is ulva. And ulva is what I kind of call the signature seaweed of the green category. And it too is only 2 cells thick, distromatic, and but it's not stretchy. And you will find this. This is absolutely everywhere. It's in the higher intertidal zone than probably the Pyropia or Nori. But it is pure green because it has only chlorophyll in its chloroplasts. And it is the green seaweeds that migrated up onto the near shore and gave rise to and evolved into the terrestrial plants, terrestrial vascular plants. So it's the chlorophyll of the green algae that kind of gave us the chlorophyll and the green of our terrestrial landscape. And this is that great bladder chain rack, right, with its Ruprecht lithograph. And this was the image. This is a rack or a kelp. It's in the brown category. And kelp, I'll go through the categories in a minute. But the kelp, if it are the only seaweeds that have bladders. Well, sargassum has little bladders, but the browns, the kelp, the big bladders are what clue you into it being a kelp. So this was the image that gave rise to the cover of my book, The Curious World of Seaweed. It came out in 2019, and I got to design the book, and then it's a rough design, and then I worked with the art director and designer at Heyday Books, which was such a joy, and they made a really beautiful, beautiful volume. I hope some of you can find it in your local bookstore. It's been really fun to work with the designers over there. And the book is 16 chapters. Each one is about an iconic kelp or seaweed of our North Pacific coast. These are seaweeds that everyone should be able to find on their seashore or on their rocky coast. And they're each chapter, while I highlight all these pioneering women scientists in each chapter, I really try to write it as an algal-centric story, really trying to keep the organism forefronted, because I'm very conscious of the fact that we tend to center ourselves, and that's not necessarily the storytelling technique that benefits our natural world the most. So I'm going to go in here to remind us that those 16 chapters are just a tiny of 16 species, just a tiny, tiny fraction of the incredible biodiversity of the marine algae of our North Pacific coast. And you all in the Salish Sea and in British Columbia and have such an incredible amount of coastline. You have, way, way more linear miles of intertidal coastline than we do here in California. And every inch of that, it has probably, unless it's very sandy, has probably some algal growth on it. So keeping track of the algae and how it's doing is really important. So one of the great reasons we have such a rich algal biodiverse flora here in California is that we have this great upwelling event that happens in the spring. And this is the coast of Sonoma here at a place called McCarraker State Park. And this is when the spring winds pick up and they push the surface waters away. And the continental shelf is very close to the edge of the continent. So what happens is those surface waters are pushed away and this deep, nutrient-rich, cold ocean water comes up to fill that space. And that is the upwelling that not only provides the nutrients for algal growth, but of course starts the food chain that then benefits all of the organisms up to the great whales that we've been talking about earlier. And this is just a picture of some intertidal detritus, and detritus is a great word because there are many organisms that are detritivores, like the isopods that would be found in this algal rack here. And this is to remind us not to be squeamish about the seaweed and really go out there and get down into it and remember that all of the kelp flies and the isopods that are decomposing the seaweeds on the shore are incredibly important nutrients for shorebirds and other birds, especially migrating birds. Think of the protein that is there for bird populations. I also show this because it's just got all this color and texture in it, and it's to kind of highlight the greens, the reds, and the browns and the textures that you'll find in the intertidal zone. So our seaweeds come in 3 categories, and these are the evolutionary lineages of the marine algae. So the green, as I mentioned before, here represented by the ulva here, is purely green because it has just chlorophyll in its chloroplast. So chlorophyll is very good at collecting red light. And we're pretty, we don't think about our daylight as being very specific, but it is actually quite specific. Daylight is very heavy in the red spectrum. And so the green chlorophyll is very efficient at collecting red light and reflecting the green back to us. But the red daylight that we know and love is not necessarily the spectrum of light that will penetrate the density of the ocean. So the red seaweeds here broke off from the greens many, many, many millions of years ago, perhaps maybe even 1 billion years ago, long ago, and evolved 2 accessory pigments in their chloroplast, a red pigment and a blue pigment. And those red and blue pigments, which are good at collecting different wavelengths of light, they combine to make this array of pinks and purples, magentas that you find in the red category of seaweeds. And then many, many millions of years later, a much later evolutionary event gave rise to the brown seaweeds. And the brown seaweeds have a brown accessory pigment that combines with the chlorophyll, the green chlorophyll, to make this array of browns, olive colors, golden colors that you find in the brown category. So in my chapter, I have a whole chapter on color in this, and I use as the iconic species, I talk about Maziella volans, which is, not Maziella volans, this is Maziella splendens. And Maziella is this wonderful large-leafed red seaweed that you find in the low intertidal, and it is the seaweed that might be called a rainbow leaf because it often has this very iridescent feel to it when it's in the tide pools. And that iridescence is just an artifact of some of that it has, like abalone, many different surface layers that the light, when it's wet, is refracting off of. But when you dry it, it goes to this perfect purple color. And that's because the red and the blue pigments are coming together in kind of equal amounts. So I have a whole chapter on color. And now I want to talk to you about bull kelp, or Nereocystis luciana. And the Salish Sea was traditionally incredibly packed with bull kelp, to the extent that mariners had a really hard time navigating through some of the channels up in the northern part of the Salish Sea as you moved your way up on the east coast of Vancouver Island. The temperature of the Salish Sea has been increasing a lot, so a lot of that bull kelp has been is not able to succeed as well in the warming oceans. And this is something that Chris Neufeld and a wonderful group called Kelp Rescue is doing all sorts of work around. They're based at Banfield Marine Labs over in Barkley Sound. But this is the iconic kelp of the Northern Pacific. And it is kind of my mission right now, is to tell its story across its entire biogeography. That's kind of where I'm going now. But it begins right about now. If we were diving out in these kelp beds, you would see these tiny baby Nereocystis. And these are some pressings from the University Herbarium down here at UC California. And then they grow into these large, mature bull kelp in a matter of months. So between March and July, this organism will go from this tiny little lovely baby, and you might see those on the beaches this spring, where the bladder is just about as big as your thumbnail with these golden blades, into this massive thing that you find washed up on the beaches with a big 4 to 6 inch bulb, and these blades streaming out, and a long stipe that is accumulated its biomass simply by the power of sunlight and ocean nutrients. And of course, it needs a rocky bottom to hold on to. This is what the kelp forest looks like, hopefully right about now, or in another month or so, when these bladders, these gas-filled bladders, are pulling the blades up towards the surface so that they can photosynthesize as efficiently as possible. And what's really interesting about the bull kelp is that the point of growth is, when it's in this stage down below, is along the stipe here, and is heading up towards the surface. Once the bulb, the pneumatocyst, hits the surface, a trigger is set so that the stipe stops growing, and the meristem, or point of growth, switches to between the bulb and the blades. So the blades will continue to grow through summer. But this is an annual organism. Bull kelp is an annual. So the seaweeds do come as perennials or annuals, and so the bull kelp has to reproduce before it is kind of picked up by winter storms or is washed ashore. Now, bull kelp can have kind of, especially in calmer places, so in the Salish Sea, I wouldn't be surprised if you saw a lot of holdover or overwintered kelp, but it still has to reproduce that first summer. So by late summer, you should see these spore patches or sori on the blades of the bull kelp. And it's really interesting. When you do find it on the beach, check the blades to see if it has these dark patches. Each of these dark patches has millions and millions of spores. And then as the summer progresses, the cells around the edge give way and create that halo, and then they actually fall away. And this is what it actually looks like underwater this was late August down here in Sonoma, where the sori have fallen away. It's kind of like your cookie dough after you've cut out your cookies. So this is just a little trio of images taken down here in a place called Van Dam State Park on the Sonoma coast. And this was taken in 2009 when there was a huge year for kelp. And bull kelp is typically extremely cyclical in kind of 3 to 4 year cycles. And there's all sorts of kelp data now from mapping about the cyclical nature of it. So when we talk about, you know, the steep decline in kelp, it isn't to use this year as your year is not necessarily the way to go. This was 2009, and it was a gangbuster year. This was 2017, when I was up kayaking in that same kelp bed, and you can see everything is much more spaced out. And this was 2017, and this was 2019 and 2020. I was up there both years, and I stopped at this same space looking over Van Dam State Park, and there's no kelp at all. And I think what's really important is for us to remember what we're forgetting. There's no way to know what is missing from this picture. It was a beautiful day. The water was clear. People were kayaking. The sand, you know, it was a lovely space. And yet, if you didn't know that this was a historic kelp bed, then you wouldn't know that there was something really really wrong with the picture. So this very steep decline in kelp in California has gotten a lot of attention, and it's actually happening now as we move up the coast. And what's happened is not only did we have this enormous couple of years in 2015, 2016, and 2014 of very warm oceans, we also had an explosion of purple sea urchins that coincided with the starfish wasting disease, and we lost the predator, the urchin predator, the sunflower sea star. So this was me snorkeling and watching these urchins pin this beautiful bull kelp that was still attached to the bottom by its holdfast, pinned to a vertical rock face, and they were eating away the blades. And so the kelp forests in many places have kind of shifted over into these urchin barrens. And you see one abalone in there. The urchin barrens are where the urchins have really taken over and eaten the kelp down to nothing. And this is kind of a stable state. The urchins can live as these impoverished urchins with no food for a long time. They become kind of these zombies. Now, all of this is in the context of the fact that in our California coast, we do not have any of the other apex predator, the other top predator that worked in tandem with the sunflower sea stars to keep urchin populations in check, and that's the sea otter. And as we go up the coast, there are all sorts of different places where sea otter are and sea otter aren't. And especially along the west coast of Vancouver Island, the very last chapter of The Curious World of Seaweed is really set up on the west coast of Vancouver Island, where Jane Watson did all of these long-term experiments tagging kelp and taking note of succession of kelp beds when otter were present, when otter for long periods of time, where they had been there for decades, in places where the otter were returning, and in places like Barkley Sound where there are no otter at all. The sea otter is really, there's many different kind of takes on sea otter. They compete with us for our, as top predator, really. So there are a lot of communities that are harmed by otter being kind of detrimental to foraging foods. But this was a piece that I made that was really an homage to the kelp forest, as I was watching in real time these kelp forests disappear on our north coast of California. But now I'm just going to go quickly, so we have time for some questions, through some colorful pictures. These are just some of the seaweeds. This is the super rosy red Weeksia. It was named after the Mrs. J.M. Weeks by William Albert Setchell, and of course I talk about her. Postelsia. So if you go out to the west coast of Vancouver Island, and if you go to Botanical Beach, which is near Port Renfrew, that is a really historic algal place. And there are beds of Postelsia there. Postelsia, which is known as the sea palm, is the end of its range is Vancouver Island, but it likes the rocky, wave crash dynamic coast, and it really is shaped like a little sea palm. And I write about this wonderful woman, Josephine Tilden, who published this book on the left, the yearbook of the Minnesota Seaside Station. And she established the first marine lab on the West Coast at Botanical Beach near Port Renfrew. If you have a chance, it's a regional park now. I can't stress enough how lovely it is and what an incredible tide pooling place it is. And I write about her when writing about the coralline algae, because the guy who named the Coralina Vancouverensis, a Japanese scientist named Yendo, he got his specimens and did his research at Botanical Beach, and he was hosted by Josephine Tilden. And he took his specimens back to Japan, and then he named the Coralina Vancouverensis, which is one of the very, very fine articulated coralline. So you have on the left, you have articulated coralline algae, and on the right, you have encrusted coralline algae. These are really important foundations to our tide pool and intertidal zones. So when you go out, notice if there's this pinky crust that kind of underlies a lot of the other algae, because the corallines actually have some kind of a chemical cue for invertebrate larval stage babies to settle on the reef, and other algae as well. So the corallines are super, and this is a variety of different articulated ones. I think the Vancouverensis is the one right at the very top there. Agarum is this wonderful holy kelp. It's one of the only kelp species or seaweed species that evolved on both coasts of North America. So as you guys move up into Alaska, you will find the Agarum. It doesn't exist down here in California, really, but it does exist in Maine, which is where I found it. And so I write about a whole chapter on that, and it's a whole chapter on why these herbaria who collect specimens are so important, because it was across generations that these specimens of Agarum that started with Joseph Steller were picked up and used, were found at the Academy of Sciences in St. Petersburg and described. And then we mentioned, okay, I'm going to jump to surf grasses and eelgrasses, which was mentioned as somebody was doing some eelgrass restoration, which is so great. So the eelgrass is the Zostra marina, or Zostra marina, and it grows in the bays and estuaries. But the surf grass, or Phyllospatix, is what grows out on the rocky reef. And these are flowering plants that re-evolved back into the ocean. And they have a wonderful algal epiphyte that grows on them called Smithora. And it is this beautiful red algae whose spores will only settle on the thin blades of the Phyllospatix. You can see down at the bottom here that those are the flowers, actually. So again, the eelgrass and the surfgrass are flowers, are flowering plants that evolved back into the ocean from land. And this is what my scanner allows me to do, is to create these really vibrant and fabulous portraits. These were some specimens of eelgrass taken from the tanks of Kathy Boyer, who is doing all sorts of eelgrass restoration here in San Francisco Bay. And I write about this incredible network of Zostra researchers that are all sharing their data. So here, these are just some of the newer work that is really part of what I'm doing now. I was asked to create a proposal for a big mural in a law school here in San Francisco. And I thought, well, it would be good to have foundational kelp on the walls as opposed to founding fathers. But the COVID came down and that whole project stopped. But these are some images called Ocean's Edge that I'm creating as all these huge fabric panels that are about to be installed in a gallery downtown San Francisco. And we're using, it's a group of 5 women artists who are all focusing on bull kelp in particular. And we're using this exhibit as a way to bring, it's a 6-month exhibit here in San Francisco, and there's going to be panels and forums on the kelp forest and kelp restoration issues all around this exhibit. So that's very, very exciting. And to work with these other 5 other artists is also really exciting. So again, here's me playing. This was the beginning of the lockdown when I suddenly was just in my studio. I had a wonderful show at this wonderful gallery last year. And this is my main focus right now is a project called Chasing Kelp. And it's really telling this bull kelp story across its range. This is my collection that is actually back over in my garage there. And along with some of my cyanotypes. I actually cast one of my kelp collections in bronze. And one of my great dreams is to do a big bronze sculpture of the bull kelp to be in a public place so that people can have a tactile experience with this organism. So I'll end here. And I'm so please be in touch with me. My email is there. I'm at Josie Islin on Instagram. And I love chatting on Instagram. Show me your finds. Again, I hope you find the books, and I hope you get out to the intertidal zone, out to collect or at least look at the seaweed. You always have to know where you are, and if you're in a marine protected area, you shouldn't be collecting, but you can take pictures and you can notice and be amazed by the seaweeds.
That was wonderful. Thank you so much, Josie. That was, yeah, beautiful photos and amazing work. We do have a couple of questions.
Sure.
In the chat. One was, how do you how do you protect your scanner from salt water when you put the seaweed in your scanner?
Well, one of the beauties of glass is that glass is impermeable. So I don't worry about the salt water. I just keep a lot of Windex over there and I clean it a lot. So it's one of the great things I found with the scanner is that it's a very resilient tool. Right.
Question, well, it's actually a bit of a comment. The heat dome of June 2021 killed a lot of the rocky seaweed species here, and so we're not seeing too much recovery yet. But I guess I don't know. Do we anticipate recovery?
Well, the last couple of years have been a pretty cold ocean. When those heat domes come and they come at low tide, they can be really devastating. I know we had huge mussel die-offs here when some of those huge heat waves came, and they coincided with some of the big low tides. Seaweeds are pretty resilient. So if we can keep the waters cold, one of the issues with the Salish Sea is that its temperature is rising really rapidly. I was talking with Chris Neufeld, who's a researcher at Bamfield, and he has done the temperature maps, both in Barkley Sound and over in the Salish Sea on the east side coast of Vancouver. So we have to do whatever, it's going to fluctuate the ocean temperature from year to year, but keeping them cold. And if the cold water's there, the seaweeds are very resilient. So give them a couple more years.
Good to hear. Another question was, what is the gas composed of in the bull kelp bulbs?
There is some carbon monoxide in there. And it creates this gas right from the start, and it just keeps making it. I know I got to spend time with Patrick Martone's graduate students who are doing all this interesting physiological work on bull kelp, like pulling it and slicing it, and they mentioned that the gas, they didn't give me the breakdown. There's definitely some carbon monoxide in there, so it's kind of deadly, I guess, if you actually break it open and inhale it. But I know it kind of is generating that gas kind of at this continuous level. It's one of those mysteries, actually, that you need these PhD students to be investigating.
I'm looking for any other questions that came in.
I love the one about the Agarum, the Colander Kelp.
Yes, that it was in the sound, but maybe not doing so well. Where was that?
Oh yeah,
Sorry.
so green urchins, right? So you guys have green urchins. We have purple urchins down here, and you guys have the green urchins. And so when so in the sunflower sea stars were decimated along the entire West Coast. There was some calculation of how many billions of organisms died in that starfish wasting disease event. So the urchin populations have not been in check unless there are otter around. It could be that the Colander kelp is being drastically reduced by the green urchins, who are just a little bit about that trophic cascade, which is that that the kelp, a fleshy kelp like Colander kelp or bull kelp or sugar kelp, is the primary producer is the herb, the bottom of the food chain, and the herbivores in the system are organisms like snails and sea urchins and abalone. And usually in a healthy kelp forest, in a healthy intertidal, subtidal kelp zone, there's so much algal detritus, so much sloughing off of algal material, and it's why the visibility might not be so well if you're snorkeling or diving, but that algal detritus that is floating in the water column is at what typically the urchin and the abalone will feed on. They are typically known as detritivores. So in a healthy system, they just kind of stay put and they wait for the algal detritus to float by, and they will snag it and eat it. But if the balance gets kind of like pushed out of whack by some of these events that we've noticed, such as the starfish wasting disease or an ocean temperature event, and there is less and less algal detritus, the urchins will get up out of their little holes that they make, and they'll go on the march searching for kelp, and they will go find the kelp and they will eat it. So this change in behavior is really interesting. There's a really wonderful newly minted PhD student, Joshua Smith, down at UC Santa Cruz, who did some really interesting studies about sea otter and urchin and kelp in these mosaics, these mosaic of kelp forest situations where the sea otter will, the urchin that are in the kelp forest have enough protein in them to kind of stay put and have enough gonad or uni in them to actually be good food source for sea otter. So otter will really only hunt within the healthy kelp forest. But as soon as you go out of the kelp forest where there isn't as much kelp, the sea urchin are impoverished. They don't have the protein that would make it worthwhile for the otter to actually go hunt them. So what that means is that in these places where that otter are good at maintaining the health of a kelp forest, but they can't go into a urchin barren. They won't eat the urchin. They know that the urchin has nothing in it. They're not going to pull up an empty urchin. And these urchin can sit out there for long periods of time in this very empty state. They're kind of a really interesting organism that way. So, but there's all sorts of kelp restoration happening. There's lots and lots of work going on. Guayhanas Marine Reserve up at the tip of Haida Gwaii is really interesting. They're doing lots of culling of the urchins there so that the kelp can regrow. We're doing a lot of that down here in California. And the Puget Sound Restoration Fund is doing really good kelp work. There's lots and lots of good work going on. So.
Wow, no, thanks for that. A couple questions here. Someone was asking whether you would consider including microscopic images of seaweed.
Absolutely, they're really beautiful. I've thought about it, absolutely, and I have collaborated with some of the researchers and professors who teach who teach phycology or marine botany, and but I'm so still kind of passionate about telling this the bigger story that those of us can recognize. But there's plenty of time. They're beautiful. The images, the microscopic images of the seaweed is amazing. Yeah.
Oh, thanks for that. We're almost at time, although do we have time for one or two more questions? Is there
Sure.
anything else going on in the chat?
Anything else in there? Yeah, lots of good comments about kelp farming and restoration. And oh, here's one about green crab impacting eelgrass beds. Oh dear. Do you have the invasive green crab there yet?
Oh, we've had invasive crabs here for a while. Yeah.
Right. Right.
Ooh, saw some baby bull kelp last week out at Tongue Point. Good. Yeah. It's really good to notice. There should be just thousands of them on the floor bed. So some of them will always be washing up.
Yeah. And is kelp farming a good plan?
You know, I think it's all over the place. I don't really take a stand on it. I think this notion of carbon sequestration, which everybody loves to kind of hop on board with, that kelp farming is good for carbon sequestration. It's like, well, it's only while the kelp is in the ocean, and naturally kelp does degenerate so quickly. It's not like a tree that, you know, can then grow and then petrify or turn into coal or whatever it is. To sequester the carbon, the seaweeds are just, the turnover is pretty rapid. So how much, if it floats into the deep ocean, then I guess it is sequestering the carbon once it detaches. But that's, I think, really still trying to be quantified in terms of a carbon sink. They're absolutely necessary for biodiversity. They can help with acidification on a very, very localized level, the kelp farms, if you kelp farm around your oyster farm. And so I think there's just so much work going on. I spend a lot of time in Maine, and the kelp farming in Maine is huge. I'm going to be going up to Alaska, where kelp farming is big. So it's definitely part of the conversation.
Cool. All right. What do you think?
How do I preserve? I'll just talk about how I preserve the color of the seaweeds from collection until I scan it. So I can scan my seaweeds either wet or dry. So I often will come in from the beach, and of course again, I'm going to really emphasize that, you know, our marine protected areas we do not collect from, but there's plenty of places, and the rack line has plenty of seaweed. You don't have to collect live seaweed. But I will bring it in. If you need to keep it for a day or so, I'll put it in my refrigerator. You don't need to keep it in water. You just need to keep it cold. I have a little sign here from one of my kids long ago: "Mom, don't forget the seaweed in the fridge." But then I will scan it while it's still wet. And then I will press it in my plant press, and I will press it not so that it adheres to herbarium paper or to a piece of paper, but so that it is a discrete object, so I can pull it off. So here is a pressing of a bull kelp. This was a little tiny baby. You can see how incredibly fragile it is. I don't know whether you can see that. I'll hold it up against me. It's super thin. That's how much water is pulled out of it. But that's from sitting in my press for a long time. Bull kelp, it takes a couple of weeks to press it. But I'll put so I have a big plant press, and that's how I press them, and then I can stick them on my scanner. And the color, and they maintain their color for forever. I go into the herbarium and see 100-year-old pressings that are very brightly colored.
Wow. So cool. Very lovely.
Thank you so much. Yeah, thank you. That was really great. Thank you, Josie.
Absolutely. Feel free to, anybody can
send you questions would be all right.
Totally fine.
Oh, good. All right. Well, anybody with any last ones? But I guess we should wrap up here. We're just after 9, and come by next week. Hope to see you for the 4th and last evening on May the 2nd. And we'll have Towards an Atlas of Salish Sea Biodiversity by Andrew Simon, and a talk about nudibranchs up close and personal with Luann Roberts. And yeah, we'll also follow up the idea about breakout rooms. Thank you for that feedback. And yes, if you have any questions, anything at all, let us know. Any last minute things to add, Liz, or is that's it? Okay, all right. Good night, everyone. See you next week.